Thank you, Harvey. In our next presentation, we shift our gears and go into some nutritional issues. Dr. Denise Ney from University of Wisconsin-Madison is going to be presenting to us. Denise has been involved in nutrition for many years, having completed her PhD at the University of California, Davis, and she's currently the nutritional discipline coordinator for the Weissman Center Maternal and Child Health Leadership Education and Neurodevelopment and Related Disorder Program. Some of you may know Denise. She's been working with GMP for several years now, and her topic today is GMP, synthetic amino acids and casein-controlled diets on osteopenia in PKU mice. Just welcome Denise.
It's a pleasure to have this opportunity to talk about bone health in PKU. I'd like to introduce you to our UW-Madison PKU bone research team. Myself, Robert Blank, who is a physician with a research specialty in metabolic bone disease. When I became interested in pursuing the influence of dietary protein on bone health in PKU, I sought out Rob's collaboration. He said yes, he would be happy to participate in our study, and this has evolved into a very rich and fruitful research study, and hopefully we'll be applying to NIH to continue the research that I will describe to you today. Sangeeta Murli is a scientist who essentially runs my research lab, and graduate student Patrick Solverson, who conducted much of the hands-on research that I'll be presenting today. Thank you, National PKU Alliance, for funding our research. Your support means much more than just the money. It also comes with a message to my research team that the research that we are doing is important to the PKU community. PKU is associated with low bone mass, referred to as osteopenia, and fractures in early adulthood. We refer to this as the skeletal fragility of PKU. Earlier this year, a study from the Netherlands was published in which they assessed bone mineral density in 53 treated PKU patients. The authors concluded that reduced bone mineral density in PKU is present from an early age onward, and it cannot be predicted by plasma phenylalanine levels. What causes skeletal fragility in PKU? The fundamental question is whether reduced bone mineral density is inherent to PKU or secondary to its dietary management. This question cannot be answered in a human PKU study because it is essential that the low phenylalanine amino acid-based diet be initiated shortly after birth to prevent cognitive impairment. To address this question, we have conducted a factorial experiment in PKU mice. The objective was to determine how the PKU genotype and the source of dietary protein affect growth, body composition, and bone development. Male and female, wild-type and PKU mice, were assigned to one of three diets: a high phenylalanine casein control diet, or a low phenylalanine diet in which all of the protein was provided by synthetic amino acids, or a low phenylalanine diet in which all of the protein was provided by glycomacropeptide, or GMP, with supplemental amino acids. The mice were fed these diets from weaning through 23 weeks of age. And at the end of the study, the age of the mice was equivalent to about a 25-year-old human. So you see we're exposing the mice to these sources of dietary protein throughout the period when their skeleton is forming. We had over 200 mice in 12 treatment groups. This is probably the most comprehensive nutritional study that has ever been done in PKU mice. And again, thank you for making it possible. We have a PKU breeding colony. I use the same mice that Carrie and Kristen talked with you about, which is a very good model for PKU. We have the PKU homozygous mice, and we also have the wild-type mice. Now, wild-type means that the mice do not carry a defective gene for phenylalanine hydroxylase, okay? It does not necessarily mean that they're particularly mischievous or ornery. We just refer to them as wild-type. Now, these PKU and wild-type mice are housed with same-sex littermates. Okay, as you can see here, the brothers or the sisters are in individual shoebox cages, and the mice are, of course, separated according to the 3 diets, which are color-coded: pink for GMP, green for amino acid, and blue for casein. And I use these colors in my data slides, so we'll see if you can remember. You'll notice in the lower graph here that there are some, it's not just Dr. Levy. You'll notice in the lower cages that there are some little dome-shaped structures. Can you see it? There's two reddish ones, and then on your right there's a gold one. Those are mouse igloos. And the idea is, if the mice get tired of playing with their brothers and sisters, they can give themselves a timeout and go into the igloo and take a break from the rest of the gang before they go out to play. GMP is a natural whole whey protein. It is produced when making cheese. Pure GMP contains no phenylalanine. However, in the process of isolating GMP from the whey mixture, and that's what you're looking at in this vat is cheese whey, and in the process of isolating the GMP, it becomes contaminated with phenylalanine from some of the other proteins that are present in the whey. Thus, foods made with glytactin, which is Cambrooke's proprietary formulation of GMP and supplemental amino acids, these foods do contain a small amount of phenylalanine. Here we look at growth based on gain in body weight. As expected, the wild-type mice fed the casein control diet, and that is the blue line. These mice showed the greatest growth. The low phenylalanine amino acid in GMP diets produced similar growth and accretion of lean body mass in both the wild type and the PKU mice. Now, this is important because bone development reflects overall whole animal growth. And whenever you see a situation when there is poor growth, there's usually impaired bone development. And for example, the male wild-type mice fed the casein diet, the mice that grew the best. You'll see those on your left in the blue line. These mice, besides having the greatest body weight, also had the strongest bones. In contrast, on your right, the female PKU mice, shown in the pink line, that were fed the casein diet. In other words, these female PKU mice were off diet. They had the slowest growth. They were the smallest mice, and they also had the weakest bones. We used some new sophisticated equipment to conduct metabolic phenotyping studies in our mice. And so each mouse would go into this sealed chamber for 3 days, during which there was continuous recording of their food and water intake and energy expenditure as determined by oxygen consumption and CO2 production. In this slide, we look at the relationship between oxygen consumption and lean body mass. It's a little complicated, but there's 2 points from this slide. The wild-type mice, both the males and the females, regardless of which diet they were fed, they showed significantly lower energy expenditure compared to the PKU mice. And the PKU mice fed casein, or those off diet, had the largest increase in energy expenditure, about 15%. It was 13% higher with the amino acid diet, and it was only 3% higher with the GMP diet, but still higher than the wild-type animals. So these data suggest that PKU mice show increased energy expenditure, especially when they're off diet, or in this case, eating the casein diet. Another way to look at energy expenditure is food intake, because a mouse will simply eat more food to match its energy needs to grow normally. And here we see, on your right, the PKU mice fed the casein or the amino acid diet. These mice needed to eat significantly more food every day in order to have a similar growth rate, in order to achieve their growth potential. In contrast, on your far right, the PKU mice fed the GMP diet. This resulted in a normalization of their food intake, so they were eating as much as the wild-type animals with the same growth. In other words, the GMP diet was used by the PKU mice more efficiently to support their growth. Well, why would food intake and energy needs be increased in PKU mice fed casein or amino acid diets? One explanation is the cost of protein turnover, that is, the energy cost associated with urea synthesis and kidney workload, and we investigated this possibility. We observed that the amino acid diet increases kidney workload in both wild-type and PKU mice. Kidney mass, shown on your left, or 24-hour water intake, shown on your right, were both significantly increased by the amino acid diet, that's the green one, compared to the casein control diet, it's the blue one, in both wild-type and PKU mice. And as you can see, the GMP diet, compared to the amino acid diet, significantly reduced or normalized this increase in kidney mass and 24-hour water intake in both wild-type and PKU mice. So what does this have to do with bone development? I said that's what I was going to talk about today. The increased energy expenditure and the kidney workload represent a metabolic stress to a growing animal. And bone development is a very costly process for a growing animal. It requires a lot of energy, and it may be impaired when that growing animal is in a stressful environment, in this case, because of the diet. But does PKU increase energy needs in humans? Interestingly, studies in PKU children also indicate that energy needs are increased and oftentimes reduced growth can occur, especially when adherence to the low phenylalanine diet is not as good as it could be. So moving on to the bone, like human studies, we assessed bone mineral density using DEXA, and we actually have a mouse DEXA unit that's on your left. It's quite an expensive little unit. You can see the platform where we anesthetize the mice and lay them down. We actually rent this from our primate center. It comes over every month to nutritional sciences, and we scan all the mice to see how they're growing. We learn both body composition, that is fat and lean body mass, and you come by to our poster, I'll show you some of that data, which I don't have here on body composition. And on your right, you can see a DEXA scan of the mouse skeleton. So at 23 weeks of age. Again, that's like 25 years of age for a human. The mice have been eating these diets all the time their bones have been developing. At this point, we put the animals to sleep, we euthanize them, and we take out tissues, lots of different tissues. But with respect to bone, our focus was on the femur bone tissue, that is the big thigh bone. And we've got 2 from each mouse, so we ended up with over 430 femurs to analyze in our subsequent studies. We did observe that the PKU mice show reduced whole body bone mineralization compared to the wild-type mice. And this is exactly what you see in human PKU. This is what you can do with the bone mineral density measurements in humans. Now, in human studies, the bone mineral density is used as a surrogate for bone strength. It's an indication. Low bone mineral density usually is thought to indicate weaker bones. However, that relationship was actually developed in postmenopausal women, so we're not so sure how well it holds in children and young adults. In our mouse study, we were able to test the strength of the bones directly by doing specific studies on the femur that we had dissected from the animals. So femur strength was assessed with a 3-point loading test. These tests were actually done in the Department of Orthopedic Surgery over in the hospital, which has the necessary equipment. And the photograph shows you the loading force being applied to a mouse femur, which is less than an inch in length. Okay. The relationship between the load or the force being applied to the femur and its displacement, or how much it bends, is shown in this graph. So when you first start to apply the force to the femur acts like a spring, okay? In other words, if you were to release the force, the femur would come back to its usual normal shape, okay? But if you keep applying this loading force to the femur, you reach a critical point beyond which permanent damage occurs. And this is called the yield point. And then if you keep applying the force, you will eventually fracture the bone, which we did, okay? But the maximum load is the greatest force that the femur can tolerate before it fractures. And so these measurements tell us a great deal about the biomechanical properties of the bone, which you can only glean from an animal study. Here we look at representative load displacement curves from PKU, that's the red line, and wild-type mice. We see that the yield load is significantly lower in the PKU mice compared to the wild-type mice, and you can see that statistically in the histogram to your right. So what this means is that a PKU mouse femur reaches permanent damage with less force being applied to it than a wild-type femur. Okay? We also see the post-yield displacement, and that's the amount that the bone bends before it finally fractures, that this is also significantly lower in the PKU mice compared to the wild-type mice, as shown in the histogram on your right. Now, this tells us that the PKU mouse femur is brittle, okay? And to envision that, think of a piece of dry spaghetti, okay? And if you were to apply a little force to that piece of dry spaghetti, those of you who have cooked spaghetti before, it breaks very easily. That's what brittle means. And when we see this characteristic in bone mechanics, it suggests to us that there is a defect in the formation of the bone matrix protein, that is primarily collagen. Okay? The fact that the PKU mice show bones that break easily, this usually suggests a defect in mineralization of the bone. And we did observe reduced mineralization, bone mineral density and bone mineral content of these PKU mice femora compared to the wild type. So far I've been talking with you about the effects of the PKU genotype on bone. I've described what we call the PKU bone phenotype, which appears to be very similar to the skeletal fragility of human PKU. What this means is that we have a good model system to understand the pathophysiology of human PKU, bone dysplasia. But now let's talk about the diet. Specifically, how the casein amino acid and GMP diets affect bone size and strength. The photographs on your left are representative photographs of cross-sections of mouse femur bones from PKU mice fed casein amino acid or GMP diet. Now the bone is the outer ring. It's kind of like the donut in the donut. And the hole is, of course, the marrow and where the blood supply occurs for the bone. Now, when you look at those three pictures, I hope you see that the cross-section from the PKU mice fed the amino acid diet, that this bone is smaller, okay? And as shown in the histogram, the amino acid diet resulted in a significantly smaller femur bone based on cross-sectional area compared to the GMP and the casein diets. Now, the GMP diet compared to the amino acid diet actually increased bone size. And this didn't just occur in the PKU mice, it occurred in the wild-type mice also. Okay? This is important because bigger bones are usually stronger bones, more resistant to fracture. And indeed, we found that in addition to having a bigger bone with GMP, that GMP improves bone strength in PKU and wild-type mice compared with the amino acid diet. As shown in the load-displacement curve on your right, maximum load is the greatest amount of force that the femur can tolerate before it breaks. And you can see in the histogram on your right, in the pink one, that femurs from wild-type and PKU mice fed the GMP diet tolerated a higher maximum load. They tolerated more force before they broke compared to the amino acid diet. In summary, PKU mice have femora that are brittle and weak. This suggests defects in both collagen synthesis and mineralization. As shown on the right, collagen is the primary bone matrix protein. It forms the scaffolding upon which mineralization with calcium and phosphorus, shown in the little dots, occurs. Bone strength is a function of both adequate collagen synthesis, the protein matrix, as well as its subsequent mineralization. We observe that GMP increases bone size and strength. The femora tolerate a higher maximum load before fracture compared with the amino acid diet. So how do we think that GMP works to improve bone strength in mice compared to the amino acid diet? We speculate that the GMP diet provides for better, more complete collagen synthesis and subsequent mineralization because the GMP diet, at least in the mouse, clearly reduces metabolic stress and provides a dietary source which has a lower acid content than the amino acid diet and also an amino acid profile which may be more favorable to collagen synthesis. I started my talk by asking what causes skeletal fragility in PKU. Is it the genotype or is it the diet? We have answered this question. That is, both genotype and diet contribute to skeletal fragility in PKU mice. In conclusion, skeletal fragility is inherent to the PKU genotype and is attenuated by a GMP compared with an amino acid diet in mice. Now, this is actually a hopeful conclusion because it suggests that your bone health is not determined only by having the PKU genotype. It suggests that you can do something about it to improve bone health, and that is following your diet and adequate exercise. Future research is needed to determine if improved low-phenylalanine diets containing GMP will reduce skeletal fragility in human PKU. So for those of you for PKU, what can you do to optimize your bone health and prevent fractures? There's two things. You can follow your low-phenylalanine diet. This will improve your overall growth and your nutritional status, which will allow the bone to show optimal development. Remember, improved whole animal growth generally means stronger bones. And second, you can include weight-bearing exercise each day. This is particularly important during adolescence when much of the bone is being formed and mineralized. It's well known that applying force to bone increases its strength and mineralization. And we couldn't do this in our mice, but this is something that all of you can encourage your children to do. Thank you for your attention.